The mangroves mortality after Hurricane Ian in Charlotte Harbor. And this is to brief the TAC about the mangroves recovering and the monitoring efforts across the Charlotte Harbor after extensive damage from the Hurricane Ian. And the connection is because we're fish and wildlife and habitat restoration, protect, restore, and monitoring the history of habitats. That's the connection. Thank you so much, Craig, Casey, and we'll listen. Be ready for you.
Yes, thank you. My name is Casey Craig. I work with FWC within the habitat section. So I am focusing specifically on coastal wetlands. That's my area of research, which encompasses salt marshes, oyster reefs, and mangroves. And so today I'm going to be talking about some of our research ongoing in the greater Charlotte Harbor area, looking at mangrove mortality after Hurricane Ian. Next slide. So in case anyone needs a reminder or was lucky enough to forget, Hurricane Ian made landfall in September 2022 in the Charlotte Harbor area as a Category 4 storm. In the image on the right-hand side, you can see the path with the category overlaid on those little dots. And so you can see in that image that Hurricane Ian was a Category 5 storm right before it made a landfall. And so the conditions brought to this area included a 10 to 15 foot storm surge and up to 27 inches of rain, depending on where you were. In addition to sustained winds of 150 miles per hour and obviously even faster wind gusts. So just to give you an idea, again, I'm sure most of you have lived through this, so you don't need a reminder, but the image on the left-hand side is from the town of Matlacha. And so you can see what damage was done to the gray infrastructure there. So imagine the conditions experienced by the mangroves. Next slide. So when a storm comes, for the purposes of this talk, we're going to classify the stressors that they bring to mangroves as short-term and long-term. And so short-term storm stressors are going to be things like branch or trunk snapping, tree uprooting, which is when the whole tree falls over, defoliation of canopy, as you can see in the top image, as well as sediment erosion. Whereas long-term stressors are going to be things like a storm surge deposit, which is essentially marine sediment that is carried in with the storm surge and then is deposited on these islands, these lands. In this case, these are mangrove islands. And with the mangrove specifically, the storm surge can be harmful because it's made up of this really kind of slick carbonate mud or sand, and it smothers these structures on the tree roots called lenticels. And so these structures in the tree, or these lenticels, are essential for the tree to do gas exchange so that they can get the appropriate gases to their below ground biomass for respiration. We also have sulfide toxicity, which comes with altered hydrology. So with the storm and the storm surge and all this rain dumping, you do have altered hydrology from the storm, which is largely higher water levels than normal that are sitting in these areas where they're not normally. You would have a normal tidal flow. And so this can lead to sulfide toxicity because it changes the way that the microbes in the soil respire. And so sulfides are toxic to all living things. But as I mentioned, with the altered hydrology, this can be storm related or non-storm related. And so just a little bit of background about mangroves. Next slide. Is that a natural mangrove forest, they're very happy when they're inundated about 30% of the time or less. And so a lot of our mangroves in Florida and really in a lot of places are currently impounded from roads. And so when we're building these roads or berms, we're cutting off the tidal flow, which results in stagnant water, causing mangrove stress and mortality, or sometimes what we can call, or what we refer to as a mangrove heart attack. And so in the image on the left-hand side, you can actually see this. And so that is an area in Pine Island Sound that has been impounded by roads on all three sides. And in the middle, in that sort of red outline polygon, that was where the mangroves have completely died off in the middle. And so what I'm trying to emphasize here is that hurricanes are going to compound any stress or in impounded mangroves that are, they already had the stress from impoundment. When a hurricane comes, it's sort of compounding all those factors and increasing the magnitude of stress that these trees are experiencing. And so when that happens, the trees can sort of give us signs, if you will, visual cues. And so frequently you'll see discolored water. And again, as I mentioned, this kind of leads to the change in hydrology. It changes your microbial respiration. It leads to sometimes bacteria growing in the water, changing the color. It just looks like it's not supposed to. In our black and white mangrove species, we will see epicormic growth, which is when you have leaves growing out of the trunks rather than out of the branches. And as well as adventitious roots, which are when the roots are actually growing from the trunk tissue rather than the root tissue. And then in our red mangroves, we will see fungus galls. So next slide. When Hurricane Ian passed, this sort of gave us an opportunity to examine how storms or short-term damage and the presence of existing long-term stressors can impact the rate of recovery in mangrove forests. So we specifically targeted 4 areas within the greater Charlotte Harbor region, which you can see in the map on the left-hand side. That includes Gasparilla Island, Catfish Creek in the Cape Hayes area, Matlacha, and Pine Island. In that map on the left, that dark green color is going to be where mangrove extent is current. And that red line is where the path of Hurricane Ian came through the center. But it's important to keep in mind that because this was such a large storm, that all of these sites were actually within the eye wall of the hurricane, and so could be presumed to experience some of the harshest conditions brought from the storm. So at each of these 4 locations, we would establish 2 plots that are paired. These are 10 by 10 monitoring plots. One is in the fringe of the mangrove forest and one is in the basin. Next slide, please. And so at each of these plots, when we go back to monitor, we're collecting a large suite of metrics, which are kind of summarized as geological, biotic, and hydrologic. Some of our geological metrics that we look at are storm surge deposit thickness. As I mentioned earlier, this is going to be of interest because it can smother the trees and impeding gas exchange. We also look at soil shear strength for evidence of peak collapse, as well as elevation using an RTK GPS. Some of our biotic measurements include the DBH of trees, or diameter at breast height. So how large are the trees? Signs of tree stress, which are the ones that I pointed out earlier, those things like adventitious roots, epicormic growth. We would also record trunk snapping, branch snapping, and sort of provide like a rough grade of that. We would also look at tree mortality. Is the tree alive or dead? A tree would be considered alive if it had any live foliage on it. And then we also looked at seedling and sapling composition and density. This is particularly pertinent to the recovery and looking at composition of mangroves before and after the storm. And then also canopy cover. As we mentioned, defoliation is a big thing that happens and can stress trees out. In our hydrological suite, we're looking at hydro period. So we installed wells and water level loggers in each of our sites so that we could monitor the hydro period better. And we also monitored pore water sulfides in each of these plots and the salinity and depth of any standing water. So the ones that are the metrics that are sort of boldened in each of those boxes, storm surge deposit thickness, tree mortality, and pore water sulfides are going to be the ones that I'm sort of picking out to focus on for the rest of this talk, because you see we collected quite a bit of data. Next slide. And so when we dive into this data, we start looking at tree mortality first of all. So just to orient you, this is that same map that we saw before, but I just have placed these plots of tree mortality over time in each of these respective locations. So each plot has a set for fringe and basin, and then the colors correspond to the time period after the storm that we did monitoring. And so something that you'll notice here is that we did reach 100% mortality, or the mangroves reached 100% mortality in plots in our 2 of our basin sites, particularly Matlacha and Pine Island. And so this is kind of interesting because specifically with Matlacha, you can see that there's a distinction between the magnitude of mortality in the basin versus the fringe. And so when we look at this data set as a whole, not just the Matlacha data, but all of our tree mortality data, we do find that mortality is much higher, is statistically significant. It's higher than it is in the fringe forests. And so, next slide, please. When we try to look into this a little bit further, as I mentioned, we saw that tree mortality increase in all plots, but the magnitude is also very different. So when we look at the level of impoundment of our sites, which were purposely selected to have a varying level of impoundment, we see that the tree mortality is higher in areas that had more level of existing impoundment before the storm passed. And so that image that I showed you before of that mangrove die-off where the roads impounded the forest, and so you have that just sort of die-off in the middle, is actually nearby one of our areas where we chose to establish plots, and that's in Pine Island. And so that's actually where we see our highest level of mortality. Next slide, please. When we try and look into this a little bit further to help explain the differences in some of those mortality, looking at storm surge deposit, we were only able to find storm surge deposit in the Matlacha site. So that was on average 4.6 inches. So you can actually see that storm surge deposit in the image on the left, that very obvious sort of gray mud on top of the very carbon dense peat that we typically see in mangroves. And then, so that was a 4.6 centimeter deposit in the fringe of Matlacha and a 3.5 centimeter deposit in the basin of Matlacha. Something to note, because storm surge deposit is like the stress caused by storm surge deposit does take a long time to monitor. We can look at some research done by a fellow lab mate of mine, Kara Radabaugh, who published some data from 10,000 islands after Hurricane Irma, looking at mangrove recovery. And they found that mangroves in the 10,000 islands that had a larger storm surge deposit actually experienced higher or more delayed mortality. But it is also worth noting that not all impacts from storm surge deposit are negative, because storm surge deposits do import nutrients and they can help elevation keep pace as sea level rises. Excuse me, next slide. So diving further into our sulfides data. So again, we're looking at pore water sulfides within each of these plots. And just a reminder, sulfides are toxic to all living things, but they are toxic to mangroves and have negative impacts on plant metabolism and physiology. It's just at the certain levels that the stress is magnified. So in the plot on the left, we're looking at our forest type, fringe and basin again. And on the y-axis, we're looking at sulfide concentration. And so we can see that the sulfide concentration is higher in the basin than it is in the fringe, though this isn't a statistically significant distinction yet. If we visualize the data a little bit differently, looking at the plot on the right-hand side, we're looking at sulfide concentration on the x-axis and recent tree mortality on the y-axis. And we do find that there is a significant positive correlation between tree mortality and sulfide concentration. Something else that's worth noting regarding sulfides, we did not originally incorporate sulfide, pore water sulfide measurements into our suite of metrics until about, I think it was 13 months post-storm. So we have 2 rounds of data of sulfide metrics collection. And so with that small amount of data, we were able to pick up that signal amongst the noise, which is helps to explain some of the mortality that we're seeing. Next slide. So to sum about the things that we've been talking about, Charlotte Harbor mangroves are still experiencing delayed mortality from Hurricane Ian after 16 months. It takes a long time for mangrove forests to recover. And the image on the right-hand side, images on the right-hand side, that top image is actually the Gasparilla Island Basin site 3 months after the storm passed. And the image below is that same exact site from the same perspective, but 16 months post-storm, so almost a year later. And so you can see that the understory is coming in. However, we are still seeing some impacts from the storm. And so the magnitude of the mortality that we're seeing is largely influenced by the existing stressors coupled with the impact from the storm. And so mangroves that had more long-term stressors are experiencing higher delayed mortality after the hurricane, or they're having a harder time recovering. We did only find a storm surge deposit in Matlacha. This is probably something worth looking a little bit into further, and we do have some collaborators at Villanova and Pyeongchang Technical Institute in Singapore who are looking at how that storm surge deposit is getting reincorporated into the soil. So we look forward to that data. And in terms of us, we are going to continue monitoring through 2024 as funding allows. As I mentioned, we're 16 months post-storm and we're still capturing the very early parts of recovery. So long-term stress does require long-term monitoring. And so we plan to do that moving forward. Next slide. And I would just like to acknowledge funding sources and site access, of course, and then also, again, mention my collaborators who are also working on some of that geologic side of the storm surge deposit work. With that, I'd be happy to take any questions, and I also have my email there if anyone would like to reach out about anything else.
Awesome. Great. Thank you. We got plenty of time for questions. Anybody? Yep. Let's start with John back there.
Was there a survey of the kind of acreage of mangroves that was lost by Hurricane Ian? It looks like a lot, just anecdotal.
So I'm not aware of any exact study looking specifically to quantify the acreage of loss. However, there was some of the vegetative index, the NVDI, that took imagery after Hurricane Ian. And so you would be able to compare that imagery, the vegetative index data pre-storm and post-storm. So it is possible. I'm just not aware of if anyone has done that already.
All right. Oh, wait a second. I have a question in the online. Mark Schramm. Mark Schramm, we're ready for your question.
Ernesto. Hi, Ernesto. Can you hear me okay?
Yep.
Yes.
Okay, thank you. I'm sorry I'm not there in person. I have electrical issues at the house, but running on a generator. Casey, this is a great presentation. Thank you. My question is real brief. So I work with NOAA, National Marine Fisheries Service. Can you say that the mangrove impacts are kind of lateral equally amongst, say, red, white, and black mangroves, or did you kind of see more of impacts with different types of mangroves, if you have time to answer that now?
Sure. Yeah, absolutely. And that's a great question. So in short, the impacts experienced between our red, black, and white mangroves are not all the same. And that's largely because of the natural zonation that is experienced by mangroves. And so your red mangroves are much more accustomed to being more in that tidal zone, whereas your white mangroves, which are further upland, are much less accustomed to being completely saturated, like say having like a foot of water above them. Because of that, the impacts experienced are, or the magnitude of the impacts experienced from the storm are going to be different. And that's also why we tend to see more mortality in our basins. So something that I should have mentioned, a basin forest is a little bit different than a fringe forest in terms of the composition of the type of mangroves there. So a fringe forest is predominantly red mangrove habitat, which is what we would expect to see, again, going back to that zonation where they're closest to the water line, whereas our basin forests are more, they're mixed, but they have a lot more of our white and black mangrove species. And so they have a harder time recovering. And that is likely why we are seeing larger mortality in the basin, is because of the species that are existing there. So in general, I would say that your whites and blacks have a harder time recovering after the mangroves than your reds do, at least in our data.
Great, that's very helpful. And I appreciate your comment on the deposition of sediments potentially keeping up with sea level rise, but time will tell. Anyhow, thank you so much.
No,
Thank
happy
you
to
so
help.
much. Great presentation.
Thank you.
All right. Question here.
Yes, quick question. Are you aware of any similar efforts to monitor the area, the big gap between Mercury Bay and Pine Island Sound? Because that area was on the Jersey side of the storm. I rode it out for 9 hours and the storm flooded out friends and family. It seems like there's a big gap there. I'm wondering if aquatic reserve folks are monitoring that area.
Yeah, so I'm not aware of anyone else that's doing monitoring in this area. After Hurricane Ian passed, we talked to some of our Rookery Bay colleagues, and they were doing some other post-storm work, but just in a different location. So I don't know that there is any post-hurricane data for that more southern side of, like the San Carlos Bay, Dame Darling area sort of things. But
Yeah.
yeah, I don't think there is.
Yes, just a comment on that. FGCU is, whenever him and Brian McCart have done some work, looking at the same exact thing further south of your plot. So I was gonna make a comment that you might need to consider reaching out to them because I've seen a very similar presentation from them on this.
Okay, good. Thank you. Thank you. Yeah. We'll reach out to them for sure.
Okay, good. Yes, Mark.
Ariel first.
No, you
Go ahead.
One of the, I guess, paradigms we work on a lot is, mangroves recruit very readily. We generally don't restore them per se. So the time it's taken for this to recover, in this case, is this just the persistence of some of those sulfides in the soil? Like what is holding back the recruitment of new mangroves into these areas?
Yeah, good question. And it's really a combination of things. So the sulfides are going to be stressing out the mangroves. They're going to be impacting their physiology. Another large portion of the sort of recovery factor is the timing of propagule drop and when hurricanes happen. So hurricanes, especially our large and intense ones, tend to happen in sort of like the later months of the season. And so that tends to coincide with the propagule drop of your black and white mangroves. And so a lot of your black and white mangroves, actually, those propagules, I should say, get swept out in the storm. So the storm just kind of like takes them all out. And so there's not that stock where for the black and whites to come back, they kind of have to wait till the next round of, propagule drop. And then when those are available, we will see, your black and white mangroves start to establish more. Whereas our red mangroves, they're pretty much dropping all year round. So you will tend to see the red mangroves come in sooner compared to the others. So in terms of what's causing why it's taking so long, it's really just sort of like a summation of all the little tiny things. I wouldn't say little tiny things, but the other stressors. The road impoundment is one factor. The sulfide toxicity is another factor, especially more so for the basins. The propagule availability is another factor in terms of recovery. So in a lot of these spots, just because we've been there, we've been monitoring in like 3 to 6 month intervals since the storm has happened. And in those areas, we're not really seeing standing water as much. Like you have more of your natural tidal flow, but you don't have a bunch of water, like say like 2 feet of water standing in a basin that's been there for a long time. So it's just sort of the amount of time that it takes for all of these things to leave, the stressors to leave. So once the standing water leaves, that's going to help out a lot, but it's still going to take time for your pore water sulfides to recover because they're still saturated. It takes even longer for the soil to sort of dry out a little bit more, and they do still have their natural tidal inundation. So you're going to have salt water coming in through pore water. So in short, it's kind of like a conglomeration of all of those things, but a large factor of it is the timing of the storm and that it takes a lot of our black and white mangrove propagules out of the system, so that we then have to wait for, at least another year before we start to see them come back in.
Okay, thanks.
All right. And Daryl?
No, I thought you were gonna.
I have two quick little questions. One, were any of your 4 sites resubmerged in Debbie? And is that going to skew anything in your recovery because your juveniles got resubmerged and additional sedimentation?
Yeah,
And
that's
then the second
another, right. That's another sort of question mark piece to the puzzle. We haven't been back yet to, since Debbie has passed, to be able to see any impacts. But kind of along those lines, we've had tropical storms and things that have passed since then. So we do see some change, but It's not nearly as intense as like a direct hit from the hurricane. So I can't speak to Debbie's impacts. However, I would expect that there is going to be a little, there's going to be some, things like that. But we are monitoring in a couple months. So I should have that data in maybe like 2 or 3 months.
And then second question is, as far as post-storm, would you say there's a window of opportunity for removal of marine debris and stuff from within the mangroves where it's going to be more beneficial versus more impactful to get the stuff out later and cause additional damage or impacts by going in there grabbing stuff?
Yeah, in my opinion, I would say the debris and stuff going in to get the debris isn't going to have as nearly as much of an impact as the other things from the storm. Again, it's really like the chronic stress that really impacts the mangroves. And so, doing debris removal, that would be probably less, it's more of a short-term thing, like a short-term stressor than a long-term stressor. With that being said, I will say that we don't see a lot of debris, like human debris. We'll see wrack, seagrass, things like that, seaweed, macroalgae, whatever, all of the things out there, along with some trash. But because the basin forests are further in the forest, there's a lot of trash that actually gets filtered out. So you tend to see it more near the fringes. And our fringes actually recover a little bit better after a storm. So I would say that the impacts to doing debris removal, as long as you're not doing like major earth moving, like knocking down trees and things like that on a larger scale, pretty minimal. And you don't see a lot of debris in the basin forest just because of their geographic location.
Thank you.
Okay, you guys both kind of touched on my questions already, so that's why I was like, I'll wait and see what comes
Oh.
next. But so you're testing for all those parameters still? Is there like a timeline on how long the sulfides will like decline? Or is there like a model for that? And then that's kind of, and like where I'm kind of going with that is that in terms of like, where do we step in and start doing restoration versus seeing how recovery is going to happen?
Right. I'm not sure of sort of like the decay of sulfides. I guess you could say it's not really a decay. It's really just a change of the microbes in the soil. And so that relates to like electron availability, if we're getting into like the electron cascade of things. So in order to fix, quote unquote, or try to remedy the sulfide issue, it's going to be something that the microbes have to do. And so I think the way that we do that, I really don't know. I'd like to look into the literature a little more, but the way that we do that is going to be probably largely with the water. The inundation, getting like the tidal flow getting back to more of what it's naturally supposed to be. So in terms of timeline for when we might expect sulfides to go down a little bit, I'm really not sure yet. This is also very new in terms of using pore water sulfides to monitor stress in the field because pore water sulfides are sort of a volatile compound. And so they can be hard to measure in the field. And so we have thankfully found a kit that we can use in the field to get, right in situ measurements, or not right, but in the moment C2 measurements, if you will. But in terms of a threshold, in terms of like, decay, like when do we need to intervene? In general, mangroves don't need a lot of intervention in terms of restoration. If they're going to need any restoration, it's probably going to be hydrologic. And for that, we're really going to need probably like at least 2 to 3 years of data would be my guess, so that you can really monitor the hydro period. Something else that's worth mentioning is I haven't looked into our hydro period data yet. So that's sort of a big piece to this puzzle that's just not included in this presentation, again, for timekeeping purposes, but that should help to give us some information about that.
Excellent. Well, thank you so much.